All things health and fitness, this is the Army Gymnastics Podcast.
The Army Gymnastics Podcast.
Discover a fitter, healthier you at armygymnastics.com.
Welcome back to the Deep Dives. This is where we, cut through all that information overload, try to bring you the most crucial, distilled insights from a whole stack of sources. Today, we're plunging headfirst into a topic that really touches, virtually all our lives, often without us even realizing it: artificial sweeteners. We're going to try and unravel a dietary mystery that might be quietly brewing in your fridge, maybe your pantry. Are those sugar-free choices you make every day having an unintended, maybe long-term impact on your most vital organs, like your brain, your heart? Our mission for this deep dive is, I think, incredibly important. We want to explore what science truly says about the potential links between these non-sugar sweeteners and something as serious as stroke. It's definitely complex, often debated, with studies that, at first glance can seem to contradict each other. But we're here to unpack the latest research. We'll take you on a journey from, the tiny molecular mechanisms happening inside our bodies, all the way up to the vast oceans of population-level data. The goal is pretty simple: to leave you feeling genuinely well-informed, equipped with the surprising facts, maybe the nuanced understanding you need to navigate your daily choices.
Indeed. And this isn't just a fascinating field, it's one that's constantly evolving. New research is always shedding light on these long-standing questions. We've synthesized a wide array of information for you today, drawing from recent, really cutting-edge scientific journals. This includes detailed analyses of how these substances might interact with our bodies at a fundamental level, almost, you could say, atomic level. And we're putting that right alongside broader epidemiological studies that track long-term health outcomes across thousands, even hundreds of thousands of people. It's really about connecting the microscopic to the macroscopic to truly understand the full spectrum of potential effects.
Okay, let's unpack this then. So maybe to kick us off, let's define exactly what we're talking about when we say artificial sweeteners, or, as they're also known in the scientific literature, non-sugar sweeteners, NSS for short. These aren't your typical kitchen sugar, right? Instead, they are high-intensity sweeteners. They provide a taste far beyond regular table sugar, but with minimal to no caloric contribution. Think about substances like aspartame, zolfane K, sucralose, even some more naturally derived ones like stevia glycosides, often found in products marketed as natural low-calorie options. They're absolutely everywhere, just woven into the fabric of our modern diet, found in literally thousands of products you encounter daily, from your morning sugar-free beverages and low-calorie desserts, to chewing gum, even many pharmaceuticals, and energy drinks too. You'd be hard-pressed to find a processed food aisle without them. And just to give you a sense of how potent these are, let's take aspartame. It's approximately 200 times that of sucrose. That's regular table sugar and sweetness.
200 times, yeah.
That's a staggering difference, isn't it? Imagine needing just a tiny speck to get the same sweetness as a spoonful of sugar, yet it offers minimal caloric contribution. It's this seemingly magical ability, delivering intense sweetness without the calories, that made them so appealing and led to their widespread adoption in the first place, especially for people looking to cut down on sugar.
And that initial appeal, as you point out, was incredibly strong, almost revolutionary at the time, really. The main rationale for introducing and widely adopting non-sugar sweeteners was precisely that: offer the sweet taste without the caloric load of sugar. This made them particularly attractive for individuals managing conditions like diabetes, where blood sugar control is paramount and excess sugar is a real concern, or just for those trying to manage their weight. The promise was clear: enjoy sweetness, avoid the perceived downsides of sugar. However, as with many advancements, the deeper we look into their long-term effects, the more complex the scientific picture becomes. As our scientific tools and understanding evolve, debates over its safety have persisted for decades, particularly about those long-term health implications that might not be immediately obvious.
And this is exactly where the controversy, especially around aspartame, really takes center stage. Because you see, we've witnessed quite different views from major health organizations, which understandably can be pretty confusing for anyone trying to figure out what's safe. For example, you've got the U.S. Food and Drug Administration, the FDA. They've set an acceptable daily intake, or ADI, of 40 milligrams per kilogram of body weight for aspartame. Now, this ADI represents the amount that can theoretically be consumed daily over a lifetime without appreciable health risk. And based on their evaluations, the FDA has consistently deemed aspartame safe at recommended doses. That's a pretty clear stamp of approval from a major U.S. regulatory body. But then, You fast forward to 2023, and the World Health Organization, the WHO, through its International Agency for Research on Cancer, IRC, they made a different kind of classification. They classified aspartame as a Group 2B potential carcinogen.
Group 2B, yeah.
Now, to clarify what Group 2B means, it signifies possibly carcinogenic to humans. This classification is often based on, well, limited evidence in humans or maybe sufficient evidence in animal studies, but not necessarily strong evidence in both. So you have one major authority, the FDA, saying it's safe at recommended doses, and another, the WHO's IRC, saying it's a possible carcinogen. That's a real head-scratcher, right? Creates a lot of public confusion.
It absolutely is. And what's fascinating here is how these seemingly conflicting classifications aren't necessarily about who's right or wrong. It's more a reflection of the inherent complexity of scientific evidence, the different mandates these organizations have, even the different types of studies they prioritize. The core of this ongoing debate often stems from the limitations of early animal studies. You see, in these initial studies, extremely high doses of aspartame were often given doses way beyond what a human would typically consume.
Right, orders of magnitude higher sometimes.
Exactly. Which makes the results incredibly difficult to extrapolate to long-term, low-dose human consumption. It's like, as you said, trying to understand a single drop of rain by watching a hurricane. The scale is just completely different. This really highlights why moving beyond just broad observations and truly understanding the molecular mechanisms, how these substances actually interact with our biology, is so crucial. And that, in fact, brings us very nicely to some very recent, highly relevant research that takes precisely this deeper dive into the how.
Right. So to truly grasp the potential ways these sweeteners might affect our health, especially concerning something as critical as stroke, we absolutely have to move beyond just observing associations. We need to get granular. We need to understand the underlying biology. And that brings us to a really cutting-edge study published just this month, July 4, 2025. It uses incredibly sophisticated techniques like network toxicology and molecular docking analysis. This isn't just looking at what happens on the surface. It's about delving into how aspartame might interact with our biology at that fundamental molecular level. Like tiny keys fitting into very specific locks in our cells, potentially changing their function.
Precisely. This study represents a significant step in trying to unravel the molecular link between aspartame and ischemic stroke. And just for context, ischemic stroke is a huge global health issue. It's the second leading cause of death globally, and tragically, it accounts for approximately 87% of all stroke cases. Basically, a blood clot blocking blood flow to the brain.
Depriving it of oxygen.
Exactly. So what these researchers did was quite ingenious. They started by predicting potential aspartame targets in the body, looking for those locks that aspartame or its breakdown products might interact with. Then they intersected those predicted targets with known ischemic stroke-related genes. It's in this overlap where the potential mechanisms of aspartame's influence might emerge. And through this rigorous computational process, they identified a significant overlap, 201 overlapping genes. From that pool, they managed to pinpoint 5 core targets: IL-1B, MMP9, SRC, AGT, and TNF.
Okay, IL-1B, MMP9, SRC, AGT, and TNF.
Yeah. And these aren't just random genes, they are central players in really crucial biological processes. Identifying them gives us a theoretical roadmap for how aspartame might be exerting its effects and possibly contributing to stroke risk.
Okay, here's where it gets really interesting, maybe a bit complex, but stick with this because it's quite illuminating. When these core targets, IL-1B, MMP9, SRC, AGT, TNF, were analyzed using tools like gene ontology and KEGG pathway enrichment, they started painting this detailed picture of how aspartame might actually influence stroke.
That's right.
GO and KEGG pathway enrichment, like sophisticated mapping tools. They don't just tell us which genes are involved, but they help us understand what jobs those genes do and how those jobs connect to larger biological pathways. It's like finding a new cog and immediately seeing how it fits into the whole engine. What's truly striking here is how aspartame, this seemingly simple additive, might be pulling levers on multiple critical biological systems at the same time. Systems governing everything from blood pressure to brain cell communication. Specifically, these genes are involved in critical processes for maintaining cerebrovascular health. We're talking blood pressure regulation, fundamental for healthy blood flow, and coagulation fibrinolysis balance. That's your body's precise system for blood clotting and clot dissolving. A disruption here could be dangerous. They're also involved in extracellular matrix, ECM remodeling, which is about the structure and integrity of your blood vessels, keeping them strong and flexible. And furthermore, these pathways play roles in hypoxia adaptation, how your body handles low oxygen.
Crucial instrument.
Exactly. Also, inflammation regulation, which can be protective or damaging, apoptosis, or programmed cell death, and neurotransmitter homeostasis, that delicate balance of chemical messengers in the brain. When you see this list, it becomes abundantly clear these are all highly relevant to healthy blood vessels' brain function, and disrupting them could directly contribute to issues like stroke.
And if we connect this to the bigger picture, the molecular docking analysis, which is basically a computational prediction of how strongly molecules bind to proteins, showed strong binding affinities between aspartame and these targets. The binding energies lower than 5 kilocomol indicate stable interactions, especially for AGT at 6.7 kilocomol and TNF at 5.0 kilocomol.
So a strong connection.
A very strong connection, yeah. This isn't just a weak handshake. It suggests direct modulation of these crucial proteins by aspartame. The fact that aspartame sticks so strongly suggests it could significantly alter how these vital proteins function. This computational evidence provides a theoretical basis for how aspartame might influence these stroke-related pathways. So, to elaborate a bit, the study theorizes that aspartame may contribute to ischemic brain injury through multi-target interactions. It's not just one pathway, but potentially a cascade across multiple systems, specifically potentially disrupting inflammatory responses and vascular homeostasis. Think of homeostasis as the body's natural balance. If aspartame throws that off in inflammation or blood vessel regulation, it could create vulnerability, increasing stroke risk. It's this theoretical network effect.
That's a truly detailed picture.
Oh.
So how exactly might this happen on a chemical level? You mentioned it comes back to how aspartame is broken down, metabolized in the body. When you consume it, it's not just one substance acting. It metabolizes into three main components: phenylalanine, aspartic acid, and methanol. And each of these, the study suggests, could play a unique, potentially detrimental role. Let's take methanol first. Methanol itself is then broken down into something called formate. Now, this formate inhibits mitochondrial complex 4 and induces oxidative stress.
Okay, let's unpack that.
Yeah, so mitochondria are the power plants of your cells. Complex 4 is like a crucial generator. If it's inhibited, cell energy production suffers, causing stress and damage. This oxidative stress, like rust on the cell's machinery, is destructive. And this process is linked to angiotensin-TOA-mediated vascular dysfunction, damage to blood vessels, influenced by the hormone angiotensin II.
Right.
This damage can then exacerbate RAS overactivation, making the renin-angiotensin system even more unbalanced, driving up blood pressure and inflammation. Then there's phenylalanine, another breakdown product. Phenylalanine might interfere with large neutral amino acid transporters at the blood-brain barrier.
The gatekeeper.
Exactly, the brain's protective gatekeeper. If phenylalanine messes with these transporters, it could disrupt neurotransmitter balance, the equilibrium of chemical messengers vital for brain function. And this disruption could potentially amplify neuroinflammatory responses, basically stirring up inflammation within the brain itself, making it more vulnerable. And finally, aspartic acid. This component is an excitatory neurotransmitter precursor, a building block for chemicals that excite brain cells.
Which is usually normal.
Right. But in the context of ischemic stroke, where the brain is starved of oxygen, this could aggravate neuronal calcium overload during ischemia. Too much calcium flooding into oxygen-starved brain cells leads to damage and cell death. So aspartic acid could potentially worsen that devastating cascade.
This intricate breakdown leads us to a crucial point about the renin-angiotensin system, RAS, which you mentioned. It's the master control for blood pressure and fluid balance. AGT, or angiotensinogen, is a key component, and it's already significantly associated with stroke risk due to genetic variations. The study proposes that aspartame may upregulate AGT, potentially increasing the amount of this protein. If that happens, it leads to more angiotensin II.
The vasoconstrictor.
Exactly, the powerful vasoconstrictor. It narrows blood vessels, restricts flow, increases pressure. Angiotensin II also causes oxidative stress and endothelial dysfunction, damaging the delicate lining of blood vessels. All these factors, higher blood pressure, oxidative stress, damaged vessels, directly contribute to ischemic stroke risk. Now, while this is a theoretical mechanism from molecular analysis and modeling, there is some indirect support from in vivo studies in living organisms. For instance, some studies showed that chronic aspartame exposure, 600 milligrams a day for 12 weeks, in atherosclerosis-prone mice, actually increased vascular inflammation and plaque formation.
plaque formation, right? The hallmark of atherosclerosis.
Precisely. Atherosclerosis, the hardening and narrowing of arteries, is a major stroke risk factor. So this finding, even though it's in mice, aligns with the idea that aspartame could promote conditions leading to stroke by negatively impacting the cardiovascular system.
And what about inflammation itself? We often hear about TNFA, tumor necrosis, factor alpha, as a central player. How does that fit into this complex picture?
TNFA is indeed a central pro-inflammatory cytokine, a signaling molecule with a remarkably complex, almost dual role in ischemic stroke. On one hand, high levels can be detrimental, promoting blood-brain barrier permeability, making the brain shield leaky and causing neuronal death.
Making things worse.
Making things worse. However, curiously, some animal studies suggest that moderate or transient expression might actually induce ischemic tolerance, potentially mitigating damage when blood flow returns after a stroke. It's really nuanced. The molecular docking in this recent study showed aspartame has a stable binding to TNF with that binding energy of sp4-kilocalmol. This suggests it may directly interfere with the conformation or signal transduction of TNF-alpha.
So it could physically change how TNF-alpha works or signals?
Potentially, yes. It might change its shape or how it communicates. This direct interference could alter TNF-alpha's action, which, depending on the context, could theoretically be harmful or, in some very specific scenarios, maybe even subtly beneficial. Though we need much more research there. This does lead to speculation about a potential dose-dependent effect, meaning the amount consumed could dictate the impact on TNF-alpha.
More could be bad, less could be different.
Possibly. But it's crucial to emphasize this is still theoretical, based on computational models and maybe some in vitro evidence. Much more research, especially in vivo and rigorous human studies, is needed to confirm this nuanced dose-dependent role and its real-world implications.
That's a critical point. It's so important to remember that this whole molecular study, as fascinating as it is, provides, as they say, preliminary systematic insights.
Preliminary, exactly.
The authors themselves stress that further validation is required in actual animal models in human studies. We really need to understand the synergies of these breakdown products, how phenylalanine, aspartic acid, methanol act together.
Because the combined effect might be different than the sum of the parts.
Right. And crucially, we need to clarify the specific dose-response relationships. How much do you need to consume for these effects to happen? Is there a safe threshold? Do effects accumulate? These are the key questions for future research and clearer guidance. Okay, so that was our deep dive into the theoretical how, the molecular blueprints. It painted a really detailed, if maybe complex, picture of how aspartame might interact with our biology. But theory is one thing. What happens when we zoom out? From lab bench to real human lives, do these molecular whispers translate into actual observed effects in populations?
That's the next logical step.
That's what we'll explore next. But the big picture of population health and extensive epidemiological data tell us.
That's right. While molecular studies give us potential mechanisms, epidemiological studies look at the what in real-world settings, observing disease patterns in large human populations. A particularly comprehensive source here is that systematic review and meta-analysis by the World Health Organization published in 2022. This was a massive undertaking, consolidating findings from 283 unique studies, including randomized controlled trials, RCTs, and large prospective cohort studies looking at outcomes across the lifespan. This kind of synthesis provides really crucial insights into long-term associations.
And the findings from these population studies, specifically regarding stroke and broader cardiovascular diseases, well, they are certainly worth noting. They followed large groups over long periods, tracking diet and health meticulously. For instance, let's look at the Women's Health Initiative observational study. Huge study. Involved over 81,000 postmenopausal women.
Massive cohort.
And what they found was quite striking. Those women reporting consumption of 2 or more artificially sweetened beverages per day had a 23% increased risk of stroke. The hazard ratio, HR, was 1.23.
So about a quarter higher risk in that group.
Exactly. That's a significant association to observe in such a large group. Then there's another key piece from the Framingham Heart Study offspring cohort, that legendary long-term study.
Right, following generations.
This study reported that individuals consuming at least one artificially sweetened soft drink daily had nearly 3 times the risk of ischemic stroke, an HR of 2.96.
Wow, nearly triple the risk.
3 times the risk is a really compelling number epidemiologically, suggesting a very strong link between daily consumption and ischemic stroke in that cohort. demands attention, doesn't it?
It certainly does. And if we look at the WHO meta-analysis itself, synthesizing data from 6 cohorts, specifically on NSS containing beverage intake and stroke, it found a stroke HR of 1.19. The confidence interval was 1.09 to 1.29. Now, it's important to note that WHO categorized the certainty of this evidence as low certainty.
Low certainty. Okay, what does that mean exactly?
It doesn't mean the finding isn't real, but it indicates that while there's an observed association, more high-quality research is needed to strengthen that certainty, maybe rule out other factors more definitively. Beyond stroke, that same meta-analysis also reported increased risks for other cardiovascular outcomes. Cardiovascular events, HR 1.32, so things like heart attacks or strokes combined.
Okay.
CVD mortality, HR 1.19, death from cardiovascular disease, and hypertension, HR 1.13, or high blood pressure. These all suggest a broader impact on the circulatory system.
However, and this is absolutely vital for a balanced view, the sources also explicitly mention that other studies have not observed a significant association after adjusting for confounding factors. Things like body mass index, pre-existing conditions.
Right, this is the huge challenge.
It really highlights the difficulty of disentangling pure cause and effect in complex human diets. People choosing artificial sweeteners might have different overall health profiles to begin with, right?
Exactly. Individuals who are already overweight or have diabetes or high blood pressure might be more likely to opt for diet sodas or sugar-free foods in the first place.
So is it the sweetener causing the problem, or is it that people already at higher risk are consuming these products? This is known as reverse causation or residual confounding. It's very difficult to fully account for in observational studies.
This point about confounding factors is precisely why we need to look at all study types. Observational studies, like those cohorts, are invaluable for spotting patterns in the real world. But randomized controlled trials, RCTs, designed to control more variables, often don't show the same effects on intermediate markers. For example, the RCTs in the WHO review found no significant effect of NSS intake on systolic or diastolic blood pressure.
Oh, interesting. So the controlled trials didn't show a blood pressure effect.
Not significantly, no, nor on LDL cholesterol or triglycerides, the bad fats. There was a small but significant increase in total cholesterol to HDL cholesterol ratio, which can indicate risk, but overall, the RCTs on these markers were less dramatic.
So discrepancy there.
A definite discrepancy. It underscores the difference between short-term, controlled interventions and long-term, real-world consumption, where countless other factors are at play. Correlation doesn't equal causation, and we need more research, perhaps longer RCTs, to bridge that gap.
Okay, moving beyond the heart and blood vessels, there's also the really important question of brain health. What do the studies say about artificial sweeteners and broader neurocognitive effects? Things like memory, executive function, maybe even long-term dementia risk.
This area is particularly intriguing and again shows a mixed bag of results, highlighting how the science is still evolving here. Some randomized controlled trials, for instance, found no significant effects on cognitive or neuropsychological measures like verbal learning or memory with aspartame specifically. That might suggest no immediate noticeable impact in those controlled short-term settings for some functions.
So maybe reassuring for acute effects.
Possibly for some measures, yeah. However, one specific RCT did present a more concerning finding. Participants receiving sucralose, another common sweetener, showed a significant decrease in overall memory, encoding memory, and executive functions.
Okay, so that's different. Sucralose specifically impacting memory and planning skills.
That's what that particular trial suggested, yeah. Encoding memory is about learning new info. Executive functions are those higher-level skills like planning, problem solving. It really highlights that we can't just lump all artificial sweeteners together. They might have different effects.
Even more striking, though, was a prospective cohort study looking at outcomes over a decade. This study found that consuming NSS-sweetened beverages daily was associated with an increased 10-year risk of developing dementia, HR 2.47, and specifically Alzheimer's disease, HR 2.89.
Wow, those are big numbers.
Yeah, nearly 2.5 times the dementia risk, almost 3 times the Alzheimer's risk over 10 years. And what makes this particularly noteworthy is that the association held even after they, quote, adjusted for prevalent hypertension, cardiovascular diseases, type 2 diabetes, and risk factors for these diseases.
So they tried to control for the usual suspects.
They tried really hard, suggesting a potential independent link for the sweeteners themselves. These are substantial long-term risks that definitely raise serious questions about brain health.
And if we connect this to the broader context, beyond just stroke and neurocognition, that WHO review also touched on other significant areas, like adiposity or body fatness. Here, the findings were quite nuanced. While short-term RCTs, primarily when NSS were compared with sugars, did show small reductions in adiposity.
Which makes sense if you're cutting calories.
Exactly, if they genuinely lead to calorie reduction. But when you look at the long-term prospective cohort studies, a different, often counterintuitive picture emerges. They suggest that higher NSS intake is associated with increased risk of obesity.
Wait, so the long-term studies suggest an increased risk of obesity. How does that work?
It raises that critical question about how people actually use these sweeteners. Are they truly replacing sugar to cut overall calories? Or are they adding them to an already high-calorie diet, maybe thinking sugar-free means healthy or calorie-free?
The diet soda with a large pizza effect?
Kind of, yeah. Or maybe overcompensating elsewhere because they saved calories on the sweetener. It really complicates the simple calorie reduction idea and highlights the importance of the overall dietary pattern, not just one ingredient. Furthermore, this WHO review also suggested possible unfavorable effects of NSS consumption on birth weight and adiposity in offspring later in life for pregnant women.
Effects on the baby.
Possible effects, yeah. And they also reported a 25% increase in risk of preterm birth for pregnant women consuming NSS. Now, these are early findings needing more rigorous research, especially in sensitive groups like pregnant women and children, but they underscore that the impact might extend beyond the individual consumer, potentially affecting development, adding another layer of complexity.
So what does this all mean for us then? We've journeyed from these incredibly intricate molecular interactions deep inside our bodies all the way to the broad population studies tracking health over years. It's clearly complex, sometimes conflicting. It's definitely not a simple yes or no, which frankly can be frustrating when you're just trying to figure out what to put in your coffee.
Precisely. The scientific community is actively grappling with these nuances. It's a dynamic field. On one hand, that recent molecular docking study gives us a plausible theoretical basis. It lays out how aspartame might influence stroke through pathways involving vascular tone, coagulation balance, and neuroinflammation. The idea of direct interference with key proteins like AGT and TNF provides a specific hypothesis at the cellular level, and this proposed mechanism, particularly the potential upregulation of AGT, leading to oxidative stress and endothelial dysfunction, that actually aligns quite well with some of the epidemiological evidence.
Like the link to hypertension.
Exactly. It connects to findings linking sweetener consumption to hypertension, a known major stroke risk factor. So while one study is theoretical, it starts to explain the how for what another type of study is observing in populations. It creates a more cohesive, though still developing, narrative.
Yet, as we highlighted, the population studies still show mixed results. Some find strong associations for stroke, dementia. Others, after adjustments, don't show that same significant link. Why the discrepancy? It's confusing.
It really is confusing, and it raises that critical question researchers constantly debate. Is the sweeteners themselves directly causing harm? Or are the associations driven by the underlying lifestyles of those who consume them? That whole issue of reverse causation and a tour de force residual confounding. Maybe people already at higher risk for obesity, diabetes, hypertension might be the ones choosing artificial sweeteners to try and manage those conditions. So the sweetener isn't causing the disease, it's just correlated with people already on that path.
Making it incredibly hard to isolate the sweetener's direct effect.
Incredibly hard, no matter how many statistical adjustments you make in observational studies. And we also have to remember the ongoing debate around those high-dose exposure results from early animal studies. Extrapolating those massive doses given to animals over short periods to long-term low-dose human consumption is fundamentally challenging in toxicology.
Okay, so with all this complexity, theoretical mechanisms, conflicting population data, confounding factors, what's the path forward? How do we get clearer answers? What research needs to happen next?
Future research is absolutely critical, and it needs to be multifaceted. On the mechanistic side, we need things like developing transgenic animal models for chronic aspartame exposure. These models would let researchers dynamically monitor blood-brain barrier permeability and inflammatory cytokine expression over extended periods in a controlled way, moving beyond predictions to direct observation in a living system.
Seeing how it unfolds over time.
Exactly. Simultaneously, on the population level, we need to keep conducting large-scale human studies, but designed even more rigorously to minimize confounding factors, perhaps using innovative methods tracking changes within individuals over time. And beyond just incidence, it's crucial to delineate the synergistic effects of metabolites. How do phenylalanine, aspartic acid, and methanol act together? Is their combined effect worse, better, or just different? And perhaps most importantly for practical guidelines, and for you listening, future research must clarify the dose-response relationship.
The big question, how much is too much? Is there a safe level?
Precisely. Is there a threshold? Do effects accumulate over time regardless of dose? Understanding these dose-response relationships is absolutely key for refining food safety guidelines for regulators and for individuals making personal choices. Knowledge is most valuable when understood and applied, right? This research will help us apply precise knowledge.
This deep dive into artificial sweeteners and stroke really shows us how incredibly intricate our biology is, doesn't it? And how much there still is to learn about how what we put into our bodies truly affects us. We've journeyed from that theoretical molecular docking all the way to the widespread, observed population health impacts, highlighting fascinating insights, but also significant areas needing more investigation. It's a real testament to the dynamic nature of science. Thank you so much for joining us on this deep dive into the evolving science of artificial sweeteners and stroke. We really hope this exploration has given you a clearer, maybe more nuanced understanding of this incredibly relevant, complex topic that touches so many of our lives. Keep learning, keep questioning, keep exploring. Perhaps the most interesting takeaway isn't just what we consume, but the incredibly intricate ways our bodies process even the smallest things, and just how much more we still have to learn about the long-term ripple effects on our most vital systems, like the brain and our cardiovascular network. It's a reminder, really, that even seemingly innocent choices might have profound long-term consequences, that science is only just beginning to fully unravel.
Thanks for listening. For all things health and fitness, subscribe to our newsletter today. Find out more at armygymnastics.com.